Assalamualaikum warahmatullahi wabarakatuh. My name is Aini Alfisyahri from Husbandry B. The first topic we're going to talk about is gene interaction. Gene interaction is the interplay of several genes. Gene interaction occurs when two or more allelic or non-allelic genes of the same genotype affect the outcome of a particular phenotypic character. In the gene interaction, there is an intra-allelic. What is intra-allelic? Interaction between alleles at different locus. Genes at one locus affect the expression of other locus, or genes at one locus interact with genes at other locus. The interaction between these locus will change the distribution pattern in the F2 population of a gene. The locus that suppresses or hides the action of a gene at another locus is called epistasis. The suppressed gene is called hypostatic. Interallelic events. The first one is recessive epistasis. When recessive alleles at one locus mask the expression of both dominant and recessive alleles at another locus, it is known as recessive epistasis. This type of gene interaction is also known as supplementary epistasis. A good example of such gene interaction is found for grain color in maize. Second one is dominant epistasis. When a dominant allele at one locus can mask the expression of both alleles, dominant and recessive, at another locus, it is known as dominant epistasis. In other words, the expression of one dominant or recessive allele is masked by another dominant gene. This is also referred to as simple epistasis. Third is dominant inhibitory epistasis. In this type of epistasis, the dominant allele at one locus can match the expression of both dominant and recessive alleles at second locus. This is also known as inhibitory gene interaction. An example of this type of gene interaction is found for anthocyanin pigmentation in rice. Fourth, duplicate recessive epistasis. When recessive alleles at either of the two loci or locus can mask the expression of dominant alleles at the two loci or locus. It is called duplicate recessive epistasis. This is also known as complementary epistasis. The best example of duplicate recessive epistasis is found for flower color in sweet pea. 5 is duplicate dominant epistasis. When a dominant allele at either of two locus can mask the expression of recessive alleles at the two loci, this is known as duplicate dominant epistasis. This is also called duplicate gene action. A good example of duplicate dominant epistasis is AUN character in rice. Development of AUN in rice is controlled by two dominant duplicate genes, A and B. Finally is polymerase gene interaction. Two dominant alleles have similar effect when they are separate, but produce enhanced effect when they come together. Such gene interaction is known as polymeric gene interaction. The joint effect of two alleles appears to be additive or cumulative, but each of the two genes show complete dominance. Hence, they cannot be considered as additive genes. In case of additive effect, genes show lack of dominance. There is a deviation in gene interaction. First one is polymer. The second one is cryptomery. Third is epistasis and hypostasis. Fourth is complementary. And why this has happened is because gene interaction is a pseudo deviation from Mendel's laws that does not involve modification of the phenotype ratio, but give rise to phenotypes that are a result of the cooperation or interaction of two pairs of non-allelic genes. Second topic we're going to talk about is lethal gene. Lethal gene is that in homozygous state can causing death of the individual, whereas in heterozygous state, an individual can be normal. Type of lethal genes. Lethal genes may be classified into the following groups. First one is recessive lethal. Most of the lethal genes are recessive lethal. It is expressed only when they are in homozygous condition. The survival of heterozygote is not affected. Hot color in mice, according to Chionet, Castle, and Little, the dominant allele Y is a recessive lethal and it causes death of homozygous YY embryos at an early stage of development. Second one is dominant lethal. There are some lethal genes which reduce viability even in heterozygotes are said as dominant lethals. Epiloia gene is human beings. This causes mental defect, abnormal skin growth, and tumor in heterozygotes. Therefore, they die before reaching adulthood. The dominant lethals may be produced in every generation through mutation. Third is conditional lethal. Lethal genes that require certain conditions to act. Many other organisms are referred to as temperature-sensitive mutation. Each of them requires a definite and generally high temperature to perform their action. Fifth is balanced lethal. The balancing effect between two different lethal in self-permanent stock is called balanced lethal system Muller, 1918. Lethal genes linked in repulsion paths of linkage are said as balanced lethal. They are maintained in repulsion phase due to tight linkage. Crossing over is very low. In repulsion phase, the recessive allele of one gene and the dominant allele of the other gene are present in the same chromosome. Mating between individuals heterozygous for this balance. Lethal will produce 4 types of zygotes. 1/4 will be homozygous for the recessive lethal and will not survive. Another 1/4 of the zygotes shall be homozygous for the other recessive lethal and will die. 6 is gametic lethals. Some genes make gametes unable to fertilize. The term meiotic drive is used to describe the gametic lethal. The meiotic drive can be called a mechanism that leads to the production of unequal number of functional gametes by heterozygotes. It has been found in certain men, Drosophila pseudomscura, producing only half the number of sperm compared to normal men. The next topic is multiple alleles. Alleles are genes located at the same locus and have the same or almost the same task. Multiple alleles is the phenomenon of the presence of 3 or more alleles of a gene. Usually, one gene is composed of 2 alternative alleles. Multiple alleles can occur as a result of mutation, changes in the structure of the DNA molecule. Mutation will produce many variations of alleles. Whenever there are more than 2 identified alleles for a gene in a population, there will be a series of multiple alleles. Multiple alleles in human. Multiple alleles in human are found, for example, in the ABO and RH blood groups. ABO system blood type is affected by IO, IA, and IB alleles. IO is recessive to IA, and IA and IB are mutually codominant, not overpowering each other. The interaction of the 3 alleles results in 4 variations of the blood group phenotype, namely A, B, AB, and O. People with the IA allele can form agglutinogens or antigens called antigen A on the surface of erythrocytes and form antibodies or agglutinins or anti-B in serum or plasma blood. People with the IB allele can perform B antigens in erythrocytes and anti-A substances in blood serum. The next topic we're going to talk about is mutation. The occurrence of mutation is called mutagenesis. Living things that undergo mutation are called mutagens. Mutations are random. 90% are actually detrimental to individuals or population is a species. In general, The mutation is detrimental, the mutant is lethal, and homozygous recessive. But mutations are also beneficial, including through mutation polyploid plants can be made with superior characteristics. Factors that cause mutation or mutagens are divided into three, namely chemical mutagens. Examples are colchicine and digitonin substances. Colchicine is a substance that can prevent the formation of spindle fibers in the process of anaphase and can inhibit cell division in anaphase. The causes of mutation in the environment that are chemical in nature are also called chemical mutagens. And there is a physical material mutagens. Example of physical material mutagens are ultraviolet light, radioactive rays, and gamma rays. Ultraviolet light can cause skin cancer. The causes of mutation is in the cycle environment are radiation and temperature. Radiation as the cause of mutation is divided into ionizing radiation and non-ionizing radiation. Ionizing radiation is high energy radiation, while non-ionizing radiation is low energy radiation. Symptom is mutagen of biological materials. Viruses and bacteria are thought to cause mutation. The part of a virus that can cause mutation is its DNA. Kind of gene mutation. Based on the mutated cells can be divided into two types, namely somatic mutation and gametic mutation. Somatic mutation are mutations that occur in somatic cells, namely body cells such as skin cells. This mutation will not be passed on to their offspring. Gametic mutations are mutations that occur in gamete cells, namely cells of the reproductive organs that include sperm and ovarium in humans. Because it occurs in gametes, it will be passed on to the offspring. Based on the place of occurrence of mutation can be divided into two types. The first type is big mutation and the second type is minor mutation. Big mutation or gross mutation are changes that occur in the structure and chromosome arrangement. Point mutation or small mutation are changes that occur on the molecular arrangement or DNA gene. Okay, thank you for your attention. Hope this will be beneficial for you. That's all for me. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.